The liver is the largest solid organ in the body. At a glance, the liver may look homogeneous, but in reality, the liver is a complex organ that performs many crucial functions in supporting homeostasis. Generally, the liver is thought of as a blood detoxification machine, filtering the body's blood supply every 3 to 4 minutes from two different sources: the portal vein, which brings in nutrient-rich blood from the digestive system, and the common hepatic artery, which brings in oxygenated blood. However, the liver is also involved in a variety of other functions: the production of bile, the metabolism of fats, proteins, and carbohydrates, the synthesis of proteins such as albumin, an important plasma protein, and the immune response of detecting and clearing pathogens. It also has the remarkable ability to regenerate itself. To understand how the liver is able to perform these different functions, we need to take a closer look at the diverse architecture involved. The liver is made up of repeating hexagonal units called lobules. Each hepatic sinusoid of the lobule houses an interlobular portal triad comprised of a branch of the bile duct, hepatic artery, and portal vein. It is at this level that the liver attempts to clear substances and responds to harmful pathogens before draining the blood from the hepatic artery and portal vein into the central vein. A rich cellular microenvironment is involved in this process. Hepatocytes, which make up the majority of the liver, are involved in several metabolic processes, as well as bile production. Cholangiocytes, which line the bile duct, help modify and transport bile. And hepatic stellate cells, which reside in the space of DISA, are the main vitamin A storage site. As blood flows through the sinusoids, certain cells are involved in the constant immune surveillance of harmful substances. Liver endothelial cells, which provide a barrier between the blood and the hepatic microenvironment, but allow for communication through their specialized fenestrations. Kupffer cells, which are the liver's resident macrophages and perform phagocytic functions. And other immune cells in the circulation, including monocytes, natural killer cells, B cells, and T cells. During homeostasis, the liver is able to handle this level of immune surveillance and clear mild threats. However, the liver can become damaged over time due to immune dysregulation, such as transplant rejection or autoimmune diseases, infection, such as hepatitis A, B, and C viruses, genetics, such as metabolic liver disease, and environmental factors, such as diet. Over time, such damage can result in progressive liver disease. Chronic inflammation can lead to the development of scar tissue, known as liver fibrosis. If the scar tissue damage becomes extensive enough, it can lead to cirrhosis and severely reduce normal liver function and its ability to regenerate. Damage to the liver can also lead to the development of nodules or liver cancer. In severe cases of liver cancer or progressive liver disease, parts of the liver may need to be resected or the whole liver replaced. The liver's ability to regenerate promotes hepatic tissue growth from the remaining healthy portion of the original liver or from a portion of a healthy liver transplant. During progressive liver disease, the liver populations respond in various ways. Hepatocytes are promoted to regenerate. Hepatic stellate cells contribute further to fibrosis for their role in laying down extracellular matrix. Liver endothelial cells become less fenestrated. Kupffer cells attempt to control inflammation by recruiting peripheral immune cells, such as monocytes, which infiltrate in order to reduce pathogen spread but can cause further inflammation, and natural killer cells, which activate cytotoxicity to defend against infection, but can also cause damage to healthy cells. This immune microenvironment plays a role in both immunoregulatory functions in response to injury and pro-inflammatory processes which can cause further damage. By better understanding the way in which the liver functions at the single-cell level, we can harness the diverse functions of the cells to promote healing and reduce harmful inflammatory responses throughout the whole liver.